Good afternoon to all of you. My name is Elsken van der Wal and I'm the current chair of the Strategic Program Cancer of the UMC Utrecht. And on behalf of the program, it's my great pleasure, but most of all a great honor, to welcome you at the Associate Professor lecture of Dr. Aniek Janssen. And before I give the floor to her to give her lecture, I would like to give a brief introduction. The career of Dr. Janssen has been formed by two fertile grounds, that of Utrecht and of California, USA. This already started during her master's studies in biomedical science at Utrecht University, when she, as an undergraduate student, spent half a year at Loyola Institute for Allergy Immunology in San Diego. Following her master, she started as a PhD in the groups of Professors René Medema and Geert Kops, then working at the UMC Utrecht. Her PhD studies focused on understanding the cellular consequences of chromosomal instability. Dr. Janssen showed that chromosome segregation errors cause DNA damage during cell division, and that this damage can lead to chromosomal translocations. For this work, she was awarded a Best Publication of the Year award, as the first of many awards to follow, and she was also interviewed by the journal The Scientist. Next to this topic, her PhD research looked at how to exploit chromosomal instability in anti-cancer strategies as a way to improve the outcome of our patients. To answer that question, Dr. Janssen developed intravital imaging techniques in mice to visualize chemotherapy effects in tumor tissue, and generated two conditional mouse models to study the effects of chromosomal instability in vivo. In 2012, she finished her PhD, which was awarded with honors. We would call that in Dutch cum laude. In 2013, America was there again, because she left Utrecht to go to the USA to continue her research career as a postdoc in the Lawrence Berkeley National Laboratory in Berkeley, where she joined the lab of Dr. Gary Karpen. Here, she developed new inducible systems that allow for locus-specific DNA damage induction in Drosophila tissue, which research was recognized with fellowships for its excellence and originality, and not one, even two fellowships, one by the Dutch Cancer Foundation for basic research and a Rubicon postdoctoral fellowship from the Dutch Research Council, NBO. This research also made her an expert in genome stability, not only in mammalian, but also in fly system, and caused a long-standing and, as she told me, still deepening love for Drosophila as a very elegant and important research model. Fortunately for Utrecht University, she returned to the UMC Utrecht in 2019 to become an assistant professor at the Center of Molecular Medicine, CMM, here at the UMC Utrecht, where her research program centers on how misregulation of chromatin-specific repair mechanisms affects disease progression, and how these defects can be exploited in anti-cancer strategies. Again, her research quality was nationally and internationally acknowledged, as she received a starting grant of the European Research Council in 2019 and a so-called VD grant of the Dutch Research Council in 2021. Dr. Janssen is a regularly invited speaker at national and international podia, as well as an organizer of many, many conferences herself, and she has a great affinity with teaching and training. And luckily for us, she is also a member of the Onco Career Board of the Strategic Program Cancer. In 2021, she was appointed Associate Professor at Utrecht University. And today, we celebrate this appointment, and we honor her by giving her the lecture, time to give the lecture, and we all look very much forward to hear her lecture. Again, it's my great pleasure, but most of all a great honor, to give the floor to Dr. Aniek Janssen. Aniek, the floor is totally yours.
Well, thank you, Elske, for this very kind introduction. And thank you everyone at home or in the lab for following my lecture with the title Guarding Our Silent DNA, Why and How. So our bodies consist of 37 trillion cells, which is 37 million times a million cells. This is a lot of cells. And they all need to function together to keep our body healthy, to keep our organs functioning. But what instructs a cell to behave in a certain way? Well, that information you can find in this little round structure here in the cell, which is called the nucleus. And in the nucleus is where our DNA resides. And this DNA acts as a code. It contains of these different nucleotides called A, C, G, T, and the code is usually called a gene. And one gene can is a blueprint for one protein. And yeah, you can imagine that there's a lot of DNA in our cells, and that also means that there are a lot of proteins in our cells. In total, it's estimated that there are 20,000 to 25,000 genes, which also means that in theory we could have 20,000 to 25,000 proteins acting in our cells. So this is a lot of DNA, and this DNA is divided over 46 chromosomes in total. We get 23 chromosomes from our biological mother, and we get 23 chromosomes from our biological father. And if you nicely place these chromosomes, this is a picture of the chromosomes of one single cell, and using a nice technique, we can place these chromosomes in this order. And you can see here, like chromosome 1, chromosome 2, 3, etc. And there's always one copy from your mother and one copy from your father. Well, usually these chromosomes are not this nicely placed in the nucleus of the cell. They're usually unwound. And in total, these chromosomes cover 2 meters of DNA when unwound. And this all goes together into this small nucleus of around 12 micrometers. So yeah, you can imagine that this is kind of like a ball of wool. You have all these untangled chromosomes. You can see here, like every color is a chromosome. And they're like, yeah, packed together. But you will always have parts of your DNA that will be more compact, and some parts will be more open, some parts will be more entangled. And that's actually exactly what you see if you look at this really high resolution image of the nucleus. So this is the outside of the nucleus. And here in this image, researchers did a DNA stain. And you can see you have like kind of a grayish, whitish color. And that's what we call euchromatin, which has a more open DNA structure. And we call that active DNA. Now, on the other hand, we have heterochromatin. And that's these black structures, which really means that the structures are more compact. And we call this silent DNA. So what do I mean with active DNA and silent DNA? Well, with active DNA, I mean DNA that actively produces protein. And with silent DNA, I mean DNA that does not, it does contain the code for the protein, but it does not produce it. So when will this happen? Well, you can, for example, compare a cell from your heart and a cell from your skin. You will need different proteins in your heart cell than what you would need in your skin cell. So in your heart cell, this DNA might be active, producing protein X, whereas in your skin cell, this DNA will be silent, not producing protein X. And this is how you can get all these different cell types to develop and eventually function in your body to form tissues and have a functioning, healthy body. So yeah, euchromatin, the active DNA, it has been studied quite extensively over the past century, I would say. Silent DNA used to be called junk DNA. Researchers often thought, well, it's silent, it does not lead to production of protein, it's compact, why do we really care? Well, in the past decades, we've definitely learned to understand that this silent DNA is really essential for the health of our cells as well. First of all, you don't always want to have proteins being produced in every cell. I mean, you can imagine that that will cause problems. And then on the other hand, there's also one thing that researchers have found, and you can actually see this if you look at this image, you can see that this black heterochromatin is really covering the outside of the nucleus. And researchers have also found that this compact DNA is really also there to just maintain the nuclear structure, to keep this nice round structure and not get all the blabbing of this outside membrane. So what we wonder, or research in our lab is how are processes exactly coordinated in heterochromatin. In the nucleus of a cell, there are a lot of things that need to happen for the cell to function properly. And what happens in heterochromatin is relatively understudied. And then there's one particularly important process that we are very interested in, and this is called DNA damage repair. So our bodies are continuously being exposed to damaging sources, such as UV light or cigarette smoke. And these damaging sources, they really cause damage to our DNA. So they can cause a break of the DNA strand. Usually we have dedicated processes containing of proteins. They recognize the break and they repair the break. And you end up with a perfectly normal DNA strand. So this shouldn't give any problems. Unfortunately, sometimes this repair does not go properly. And you could compare it with this broken pipe. You could use duct tape to fix this, my dad's favorite way of fixing stuff. And you can imagine that that will not lead to a really nicely working pipe. And similar to that, in our cells, if you have DNA damage, sometimes this DNA repair is not accurate. And you get small changes in the DNA. So then what is the problem with these small changes? Well, as I mentioned in the beginning, a DNA, so a code, a gene, can lead to the production of this one protein. But if you have a mutation in this gene, you can actually end up having a cancer-promoting protein, with which I mean is a protein that now will promote cell division. So you'll get more divisions, which at the end could lead to the formation of a tumor. Or you could remove a break, like usually the cell stops dividing, it won't continue anymore. But then when you have this change to the DNA, this protein could actually remove this break and then you continue dividing the cells. So it's really important that we understand how DNA damage is being repaired. And just as an example, I'm showing you some images that we took during my PhD. So this is this healthy cell again that I was showing in the beginning with 23 pairs of chromosome, right? Like one from your mother, one from your father. This looks all pretty nice. But then if you look at the chromosomes in the cancer cell, you can see it's completely messed up. You have different chromosome numbers. So instead of 2, you have 4 here. But what I really want you to focus on is these weird structures. So here you can see a piece of chromosome 2 and copy or pasted with like probably a piece of chromosome 6 or 9. And this is all due to defective DNA repair. So if you have defective DNA repair, you get random pieces of chromosomes being fused with other pieces of chromosomes. And this can then, this can happen exactly in a gene. And then you end up having this mutated gene, mutated protein, and this could lead to cancer in the long term. So luckily, we've learned a lot about DNA repair mechanisms. These 3 scientists in 2015 received the Nobel Prize in Chemistry for the mechanistic studies of DNA repair. There are a lot of types of DNA damages, and these scientists have really spearheaded our understanding of this. But as I mentioned, we are interested in how processes occur in this compact DNA. So our question in the lab is, how is DNA repair actually coordinated in this compact heterochromatin? Because as I mentioned, heterochromatin is essential for our cells, so it's important that we also understand how damage is being repaired there. And what actually has been found is that there are a lot of DNA mutations accumulating in this compact DNA. So this really indicates that it's important that we understand how damage is being repaired in heterochromatin. So to do that, we use human cells in culture, but as Elsken also indicated, we also use fruit flies. If you look at this picture here, you're like, well, you know, a human and a fly, it's so different. But actually, they're much more similar than you might think. And in fact, 70% of all disease genes in humans have a counterpart in fruit flies. And many of the genes that are mutated in cancer were originally found in this little guy. And moreover, it's not only that these genes have been found, also many of the tissues that you find in humans have a counterpart in flies. There's many people working on the brain, and they do studies specifically in flies, or heart disease, again, in flies. So it's really a very useful model in biology. And then for us, we're also very interested in this model organism because heterochromatin, this compact DNA, and DNA repair are actually very similar between these two organisms. Here we labeled a protein that localizes, that goes to the compact DNA structures. And there are some differences, you see a little bit more in humans than that you see in flies, but really the composition, the proteins that go there are really similar. So yeah, and why do we use both? Well, the advantage of using human cells is that you can study things in a more clinically relevant setting. But the advantages of flies are, well, first of all, very fast offspring. Yeah, it takes like a female is like it comes out of your pupae, and a couple of hours later it can already lay an egg. If you compare that to the average age in the Netherlands, I think women are 30 when they have their first babies. So this already, not that I would ever do an experiment on humans, but this already indicates that flies are much easier to work with. You have much less DNA, so experiments are cheaper, faster, and what we also really like, it's an organism. You're looking at multicellular tissue, right? You have different tissues and you have an actual live animal that's, yeah, you can study your process of interest in. So yeah, what has been found using both human cells, mouse cells, fruit flies? Well, what has been found is that if you induce DNA breaks, DNA damage, and we can use this and do this in the lab, that specific, the breaks in euchromatin, so that's that gray area here again, and the heterochromatin, which is the purple area here, that the breaks in euchromatin don't really move. They get broken and they just get repaired right there. But then the breaks in heterochromatin, they move. They move all the way to the periphery of this heterochromatin domain. And the idea is that you need to move the broken DNA away outside of this compact domain so that the repair proteins can get in and can actually repair this damage. So to study this in more detail, in our lab we have fruit flies as well as human cells in culture, and we have either heterochromatin or euchromatin DNA, and using proteins that can cleave DNA, such as IC or CRISPR-Cas9, we can induce DNA damage in either the more open DNA structures or the more compact DNA structures. And then one thing that we could do is, for example, do live imaging, see how this break moves through the nucleus of the cell. So here in purple you have this compact DNA structure, and then in green is a protein that binds to DNA damage. And you can really nicely see that this break is induced right in the heterochromatin domain. And if we follow this over time, we will also see it move. So this is one of the things that we can do with these systems. So what we have found using these systems is that actually if you induce this break, so this single cut in the heterochromatin, that you need local changes here at the break site. And this is both at the DNA level, but also at the protein level. These proteins need to localize there, and this allows the movement of the break to the periphery of the heterochromatin domain. So yeah, currently we are investigating this in more detail, but I would also like to highlight a bit of the other things that we are interested in. So on the basic science part, of which which I think is really essential to move forward, is to understand how the composition of the heterochromatin domain. So which proteins bind there, and what's the structure of this heterochromatin. And then we're also interested in other processes. So of course DNA damage is one of these processes, but there are many other processes happening in the nucleus, and we also would like to understand how those are regulated. And then, of course, in the long term, we would also be really interested in bringing this basic science more to the clinic and trying to understand all the fundamental insights that we got from doing the experiments on compact DNA. Can we maybe identify a role for heterochromatin and heterochromatin changes in the development of cancer? So it is known that if you just simply look at the nucleus of a healthy cell or a cancer cell, that the chromatin structure looks very different. So this already indicates that we might want to look at those heterochromatin changes in more detail. And hopefully this will then in the long term lead to the development of new anti-cancer treatments. So I would like to end with thanking some people. This is kind of like my scientific life. So I would like to really thank René and Geert, who have been my PhD advisors and have been really excellent mentors. I also really want to thank Gary, who gave me the opportunity to do a postdoc in his lab in Berkeley, which was an amazing time. And then I, of course, would like to thank the Center for Molecular Medicine, and in particular Suzanne Boudewijn and Madelon for giving me the opportunity to hire my first PhD students. And Livio, of course, for all the help with the microscopes. And then last but not least, all my lab members that really make my work life a lot of fun. And I look forward to all your findings in the future. And the funders, obviously. And thank you for your attention.
Well, thank you, Dr. Janssen. Talking about fun, this was great fun, but it's very clear to all of us that you're a great teacher. But the way you explained this very complex material for us, like a clinician, it's very amazing that you can do that. And you had very beautiful slides. I learned a lot. I won't use the word junk anymore.
Thank you.
In relation to DNA, that's it. But you did a very good job. And I think that the way you look at it, and you introduced a new model for me, like Drosophila, and I didn't know that it was 70% equal, so it's a little bit modesty here. But I now understand your deep love for that model system. And what I'm also like very much is that you look forward to what can it do and what can it bring for our patients, because I think we need to address these kind of complexity to really find a cure or even long-standing treatment for patients. So thank you very much, and I wish you all good luck with your lab for doing your research. Thank you so much, and congratulations again with your associate professorship.
Thank you.